All right, everybody. Thank you for tuning in yet again for our next installment. We will begin momentarily in a couple minutes as we wait for people to trickle in. Thanks for tuning in. All right, everybody. Thank you again for tuning in. We'll give it one more minute to allow attendees to join, and then we'll get started. All right. Thank you, everybody, for tuning in to our third installment of what we're calling Tropical Thursdays between XPRIZE and the Natural History Museum in Los Angeles. Tonight, we have yet another exciting conversation that I'm personally very interested in. And just to get started, once again, my name is Peter Houlihan, technical lead of our XPRIZE Rainforest, a 5-year, $10 million competition to innovate and revolutionize the way that we monitor and survey tropical rainforest biodiversity around the world for conservation. And tonight we have Dr. Brian Brown joining us, and he and I are very like-minded in our entomology insect background. And Brian is the head of the Natural History Museum in LA's entomology department and curator of entomology. He has pursued an interest in insects since he was 5. And that's when he created an insect zoo in his own backyard in Toronto, Canada. And so we certainly have a lot in common in that respect, except growing up in different countries, I suppose. Dr. Brown received both his bachelor's and master's degrees at the University of Guelph in Ontario, and then earned his doctorate at the University of Alberta. After receiving his PhD, he spent 2 years as a postdoctoral fellow at the University of Maryland and the Smithsonian Institution in Washington, D.C., prior to joining the museum staff in 1993. Brown's research centers on the systematics, evolution, and natural history of phorid flies, which you'll hear about tonight, especially the parasitoid species known as ant decapitating flies and bee killing flies. So we're going to get serious tonight. He has collected specimens and conducted research in many countries throughout Southeast Asia, North America, and Europe, but especially in Central and South America. Through his work, he has discovered and described about 500 new species of phorids, and he has investigated their varied lifestyles. He has worked extensively on fossil species preserved in amber, especially from the 40 million year old Baltic amber of Europe. And so with that, we're going to get started on our conversation for tonight called Layer by Layer, the Importance of Studying Biodiversity on Every Level. So Brian, welcome. Thank you for joining us.
Thanks, Peter.
It's a pleasure. And so I think to get started, a good question based on your bio here, and for the audience to know, is what is a phorid fly?
Phorid flies are one of 160 or so different families of flies in the world. And I know flies are probably the least liked group of insects, but that's part of their appeal for me, just the sheer discovery that's involved in working with them. People have worked on beetles and butterflies to a much greater extent than worked on flies, especially the flies that I work on, which are like 0.5 millimeters to 3 or 4 millimeters long. In fact, go ahead.
That's why you got started in these, in fact, is because they were less well studied and less well liked.
Right. My supervisor, my prospective supervisor, is the great entomologist Steve Marshall. And Steve asked me what I wanted to do as a grad student. And I said, of course, I want to work on beetles. Who doesn't want to work on beetles? And he said, don't work on beetles. Everybody works on them. So I said, okay, what should I work on? He said, how about the lesser dung flies, like I work on? Maybe not. But then he said phorid flies, which I knew were interesting. At least I knew what they were. And I started getting involved in them. And I realized that that was a group that I could spend my life working on.
Yeah. And for you, I know for me, we got into insects at a similar age very early on. And for me, it was because I was just astounded by the sheer biodiversity, the number of species that insects comprise on the planet, especially in the tropics and tropical rainforests. When you started working on these, where did you first start doing your field work and collections of them?
I first started working in Ecuador. Someone told me that was the best place to start. So 3 friends of mine and I decided we would go there, and we bumbled around in the country and found some new things, but basically whetted an appetite for field work that is unquenchable, I hope. But,
Yeah.
When I was a kid, I got a little bit interested in tropical entomology, a lot interested, because I read this book called Armies of the Ant by a fellow named Charlie Hogue. And Hogue was an entomologist who went down into the tropical rainforest with a movie camera to do a series of nature videos, because they figured it was easier to teach a biologist how to run a camera than a cameraman how to be an entomologist. And Hogue was a really amazing guy. He was out there toughing it in the jungle, maybe not quite as much as he made it out to be doing it. But if you looked at the sort of role models that you could have as an entomologist before Hogue, it was sort of the effete British naturalist wearing a coat and tie with his insect net. And Hogue was out there, bare-chested with his machete, fighting through the jungle to study the army ants. And I thought that was really, really cool. And the interesting thing about that little story is that my predecessor at the Natural History Museum was none other than Charlie Hogue. So I ended up following in his footsteps.
There's a lineage there.
Yeah.
And yeah, I find it interesting that you grew up in Canada and ended up working in the tropics. And also, I mean, we've talked a little bit too about, many people think of, in terms of tropical rainforest biodiversity, we think of insects in a way because they're the most diverse group on the planet. But, many people are thinking of all the birds and the primates and the mega charismatic vertebrates. Did you ever have any interest in that as well, or were you from the beginning directly focused on insects?
I was always interested in natural history and nature in general, but I knew what I wanted to work on if I was going to be an entomologist. I mean, I went through a period when I wanted to play in a heavy metal band, and I've got the pictures with the long hair and everything.
Well,
but
long hair. But when you first went to the field, though, do you have moments of that you remember of that time, just your first experience entering the tropical rainforest in Ecuador? What stood out to you? It's very difficult. You read about it, you see photos, but actually, I mean, I think something that we can really get into tonight is field experience in the tropics. What is it actually like You know, I think a lot of times people think that it's glamorous working because you're in the tropics, and the reality is very different most of the time. And is there something that strikes you from that first experience or first time in a tropical rainforest?
Well, field work is not glamorous, as you know. It's sweaty, dirty. Um, like as not, you're walking around with bloody mosquitoes plastered to your forehead. And for me, what I remember the most about my first trip is the sounds of the tropical forest. You know, the cicadas, the tinamous,
Yeah.
all the various birds were something beyond my experience to a large extent. And when I think of my happy place, I think of lying in my bed at Tinolandia or Rio Palenque and listening to the tinamous just as the rain is starting to come in the afternoon, and
Absolutely.
howler monkeys and all those sorts of
Sounds of the forest.
iconic sounds of the tropical forest. Yeah. How about you?
Well, that certainly hits home. Yeah, I mean, for me, I first, all of those things are, to me, waking up to the sounds of the forest wherever you are, whether that's howler monkeys in the Americas or gibbons in Southeast Asia or, you know, chimpanzees in Central Africa or lemurs in Madagascar, naming all the, just the primates, you know, and
Yeah.
for us, we notice all the cacophony of insects and you know, all the species and things like that that comprise this, you know, this tropical rainforest band. But yeah, I think for me, the first time I went to a tropical rainforest was actually Costa Rica when I was 14 on a tropical biology field course. And I think that at that time, it was just something I was completely infatuated with tropical rainforests and biodiversity. And to me, it was butterflies. A lot of my early work was on butterflies and tropical rainforests. Um, but yeah, it was overwhelming. And especially the first time I went to Ecuador as well, being in the Amazon, in places like Yasuni and along the Napo River. Um, it takes a while, I think, to actually start, you know, seeing the forest for the trees, because there are so many things going on around you in many different ways that it's overwhelming. You know, you're seeing so many things, your senses are overloaded. And so the smells and the sounds and the sights, it's a lot. And so to me, you know, when I am deprived of that for a long period of time, it's hard to even describe the need to get back into the field. But as soon as you return and are immersed in that, you suddenly feel complete, you know? And for me, it's the all-encompassing experience and immersive nature of it. And certainly I think that is a component now moving into the future that's actually terrifying, as we may get into a little bit, but the removal of certain species and the collapse of the complexity of these environments. As you lose certain parts, you know, you start hearing certain sounds or you stop seeing certain species. And it's very soon after that there's a cascading domino effect of losing the whole ecosystem as a whole. Um, but yeah, before we go down into that path.
Yeah, another important aspect of it for me is the camaraderie of field work too. I have going in the field with other biologists, and
Yeah,
you know, at the end of the day, everybody sits down and spreads out what they've collected or observed throughout the day and talks about it. And it's really, it's really one of the great benefits of doing field work, I think.
totally. I would agree with that. There's a sense of community and camaraderie, but also I think after the fact as well, there's shared experiences that, like you and I tonight, have never met in person before, but we have many shared experiences of knowing what it's like to do field work, both in tropical rainforests and a lifetime of studying insects in the tropics, and knowing and understanding what's entailed the travel behind it, the difficulty, the excitement, the parasites, you know, everything that comes with it. And that I think I really value and has been interesting, especially the past 6 months during the pandemic, you know, not being in the field in the same way with your teams, has a certain loss of that camaraderie to a certain respect, but you also have the communities from before that you constantly interact with and have those shared moments. But before we go too far into the field and lose track of your forward flies, I know that you have some. I'd love for you to show some visuals.
Okay.
And both of your field sites that you're working in currently on different projects, and also give people an idea of what these insects look like.
Will do. Can you see my screen? Okay.
Yep, you're good.
Just as a little introduction, I just wanted to point out that tropical forests occupy a small, frighteningly small part of our planet. Probably around 15% of the world's surface is tropical rainforest or tropical wet forest. And we believe that about half of the world's species are found there. So that makes the world's biodiversity kind of precarious, because any loss of this tropical forest is extreme in terms of the loss of species associated with it. But it's also an opportunity. If we can do conservation in tropical areas, we can save an extraordinary number of species as well. My most recent field work in the tropics was in the center of the Amazon, near a city called Manaus in Brazil. And this is, the Amazon rainforest is the largest chunk of tropical forest in the world. And it's in great peril right now. And hopefully it's going to be there in the next 50, 100 years. We did some work with Brazilian colleagues at a site about 60 kilometers north of Manaus in the forest, where there is a tower. This tower was put up for atmospheric research, but one of my colleagues, Dr. Jose Rafael Albertino, had the great idea of putting insect traps on these various platforms that go up the tower. So this is Jose here on the left. And these traps are called malaise traps. They're like a big tent with a collecting bottle at each end. And insects fly in, make their way up to the top, and they fall in a jar of alcohol. And there's no attractants or anything involved. It's just putting these traps up, and they intercept insects that fly through. And the collections they made were truly incredible. Like I said, we had traps, or he had traps at the ground level, 8 meters, 16, 24, and 32, which go right up to the top of the forest. So the top of the forest looks like this. It's like a carpet of treetops extending off as far as you can see. And this is a kind of surface that's traversed by certain insects, just like the forest floor is. What we found was that at different levels in the forest, there were different species. And a lot of these species, of course, are associated with the trees, like these weevils, which are plant feeders. I don't know if people are aware of weevils particularly, but they're one of the most diverse families of insects, of beetles. And they have these scales all over their body that make them look like, in this case, like a little hummingbird. So they're incredibly beautiful when you look at them close up. There's lots of bees associated with the tropical rainforest, like these orchid bees. Orchid bees, as you know, Peter, are attracted to scents. They go around, they collect oils from orchids and various other plants, and they use them for attracting females and or for marking territories. And they're incredibly beautiful creatures, really easy to attract. If you want to do something to while away an afternoon in the tropics, you could do worse than putting out some wintergreen oil and seeing all the orchid bees that come by. And there's lots of hoppers down there, like this strange camelback-like one. These are plant feeders. They suck on sap. There's lots of beetles, like this flat-headed borer, whose larvae are in the tree trunks. Or this ground beetle. They're called ground beetles, but this species lives just in the canopy. They walk along the leaf surface on that surface of treetops that I showed before, and they're predators. And of course, there's lots of ants. Terry Irwin famously fogged the canopy and found huge numbers of ants, ant species, ant individuals. And some ants have scales on them like weevils do, like this turtle ant. They're really beautiful little things, although they can be annoying. But I'm most interested in the flies. And like I said, flies aren't everybody's favorite group, especially things like this flesh fly. But some flies are incredibly beautiful, like this micropesid or long-legged fly. I call these the supermodels of the fly world. And the group that I work on, as Peter said, are phorid flies. These are small, half millimeter to 4 or 5 millimeters long flies, many of which are parasitoids of other insects, which means they lay their eggs in their body, and the eggs hatch and the larva feeds and consumes the host. Of most interest on the tower, we were looking at some of these flies that attack bees. So in order to attract the flies and their bee, to attract the flies, we have to track their bee hosts. So we'd spray honey out on the undergrowth right beside the tower. And this is what happens when you do that. You can see that we get a huge number of bees coming in, drinking up the honey. And there's more than one species. There's a kind of bee in the tropics called stingless bees. Everybody who goes to the tropics knows them. They don't sting, but they're very effective at defending their colonies by biting. Okay, and now I'm going to show you one of my flies attacking one of these stingless bees. This is close up. So you're going to see it on the left-hand side in just a second. You see there's these big yellow bees. There's one of my flies. It's facing the bee. She's going to crawl up behind the bee and she's going to jump on its back. And then it curls its abdomen underneath and injects its egg layer into the bee's abdomen and lays one or more eggs. And you see she's quite persistent. It's just sitting on there. The bee doesn't seem to notice. It's drinking away the honey. And even when it gets clobbered, the fly stays on it there. So that's the kind of study we're doing. Doing this, we found a number of new species. This is one of the bee-killing flies. You can see the egg layer or ovipositor has this unique can opener-like structure on it that it uses to hook onto the bees when it's laying its eggs. This is a yellow one. There are black ones with different egg layers. And just what I wanted to point out in these few photos is how intricate these little flies are. I mean, you think of a 1 millimeter long fly, you don't think of it of having much structure, but they have incredibly detailed eyes, antennae, legs, wings, all the structures that you expect on bigger flies. And these are all useful for determining what the species are. This is a new genus of phorid flies that we found on the trip. So genus is, everybody knows the scientific name of something like Homo sapiens, humans. Homo is the genus, and sapiens is the specific name. Well, this is a whole new type, a whole new genus of phorid flies that we found on the tower. Never seen it before anywhere else. And it's this broad, flat thing that probably lives in ant nests. This is another new species. Again, look at the details of structures, all of which we can use to classify these flies. And what we found on the tower is basically equivalent to finding a new continent of phorid flies at different levels. We found things that we've never seen anywhere else, things that are incredibly rare elsewhere, probably associated with the different social insects that are in the different layers of the canopy. Okay, that's it.
Wild. That video is it's crazy. I don't know how many people are aware of you know, the predatory and the parasitic nature of many different insects that are small enough that many people may never even notice them. But yeah, a lot of the
parasitism like this was first noticed by an old codger sitting on his cabin porch in Washington, D.C. he was watching carpenter ants walking down the railing, and one of them looked like the head was a little loose, and the head fell off. And he thought, wow, that's weird. He collected the head and kept it and gave it to his friend Daniel Cocolette at the Smithsonian Institution. And Cocolette described the first ant decapitating fly based on that observation.
So some of this work, you're explaining how you started deploying your Malaise traps in the canopy, in the rainforest canopy in Manaus, in the heart of the Amazon. What really, I mean, the tower there at that site really enables you to do this type of work and access that area. But what else can you tell us from your first study? Because I think that was the first time you were deploying traps in that way in this layer of the forest. Why did you start doing that? And how has that maybe opened up the possibilities for future studies?
Yeah, I didn't do it. Jose Rafael did it. I just saw the samples when I was in another person's lab down in Brazil, and they knocked my socks off, basically, because I saw things like, what's that? That can't be. So it definitely opens up the understanding of the phorid diversity, that all insect diversity varies not only with latitude, as we know, and longitude,
Right.
but also elevation within the forest and elevation, at different parts of the continent, like in the mountains. Nobody's looked at putting traps on towers like this and other sites, as far as I know, but it would be really, really interesting to do that.
Yeah, towers definitely help facilitate that type of work. We'll have to get you tree climbing. I don't know if you've ever been.
Yeah, no, not really.
So I first started climbing trees for the work that I was doing on orchids, and for me, that I had been in canopy towers and canopy bridges and walkways before, but certainly in the tropics, especially, so much of the diversity is higher up in the canopy. And for the orchids that I was studying, well, in the tropics in general, about 80% of orchids are epiphytic, meaning they grow on other plants, they grow on trees. And so you have to get up there one way or another. But the tower approach definitely helps. I've done a few different projects where you're hoisting car batteries or all sorts of things into the canopy to light trap in the canopy or do different things. But those towers, especially the ones that you're working with outside of Manaus, help facilitate all sorts of different studies just by enabling that kind of access. Did you?
Right. Of course, people have studied canopies before for many, many years.
Yep.
Whether it's just cutting down a tree and catching stuff before it flies away, or sending up insecticidal fog into the canopy, like Terry Irwin from the Smithsonian famously used to do, and then shaking the trees to catch it. Sorry?
Or landing blimps on top.
Or landing blimps on top, yeah. So there's all kinds of different ways to study up there. I think from the samples I've found, the Malaise traps on the tower platforms seems to be the most effective for my group, but maybe for beetles. Sorry?
And why do you think that is?
Hmm.
Do you think the towers provide a little bit of a gap that facilitate maybe a flight pathway that the Malaise traps intercept?
It's possible. It's also possible that the fog and catch method doesn't work for tiny flies very well, that they waft off or don't get properly collected. I don't know. I've looked at Terry Irwin's fogging samples, and there wasn't a whole lot of interesting diversity in them for my groups.
Yeah.
So.
But you've worked in the tropics all over the world. What, for you, when you're getting back to the experience of being in the field, and there's so much more than just the collecting, as we were explaining, but even logistics. I mean, right now we're in 2020 facing a whole, I don't even know how to describe it, level of complication in terms of we can't do field work right now, or we can't do it the way that we used to. Um, what are you thinking in terms of how the pandemic right now might shape this type of work in the future? Do you have anything on the horizon in terms of thinking of when you might get back into the field, or how are you wrapping your head around this whole scenario right now?
Well, I'm using the pandemic to try and catch up with past trips. As you know, it's pretty easy to collect a lot more material than you can comfortably process
Yeah,
in a short period of time. So I've got backlogs to work on. But yeah, I need to get back into the field, not necessarily to collect more, but to continue working on life histories and understanding what these flies do. I
yeah,
mean, just knowing what species are there is one level of knowledge, but knowing that there are 30 species of bee parasitizing flies that have specific relationships with certain bees is a lot more information.
yeah,
And how those uh, relationships change over distances is interesting too. You know, I was just reading a paper about Amazonian birds and how the rivers are what segregate species in the lowlands. A lot of birds won't fly across rivers, so you can get one species or a species on one side of a river, and then on the other side something that looks exactly the same, except it's a little bit different. And rivers are associated with speciation and maintenance of species differences in the lowland tropics. So I'd really like to study that with insects as well.
absolutely. Especially there in those parts of the Amazon where the rivers are so vast.
Yeah,
Um, that certainly applies.
yeah. And Manaus is where the Rio Negro hits the Rio Amazonas, the white waters and black coming together.
Yeah.
I see a little smirk on your face. Have you read the book White Waters and Black?
No, I haven't.
No, it's a great,
I'm just smirking at the beauty of that specific
yeah.
spot, the visual, the memories, and yep.
White Waters and Black is a documentary book written about an expedition of the American Museum in the 1930s,
Okay. Yeah.
going from Bolivia to Manaus.
Okay. Yeah, I'm familiar now that you're describing it.
It's a hilarious account of poorly prepared scientists.
Well, and we can really get into that topic. I mean, a lot of what before this year, before being grounded to a certain extent, all of my work was facilitating logistics for expeditions for a variety of different institutions or NGOs or agencies. And, many people come into this, especially for the first time, wholeheartedly unprepared.
Yeah,
Uh,
wearing
and
dress shoes and things like that.
so I'm sure we have plenty of those stories as well. But yeah, do you have anything particular that stands out in that way? Anything comical or not?
Poor preparation,
Yeah.
well, or poor logistics. I mean,
Well,
I
there.
have a little story about the worst night of my life.
That's a great place to start.
Yeah, that was in Ecuador. We were going up to a station um, by mule, carrying our stuff, and we were going to walk alongside. So we loaded up our mules with all our gear, including our clothes, flashlights, money, everything. And then the mule drivers just took off up the hill to the station. And we tried to keep up, but the road was mud, about a foot deep, and we fell rapidly behind. And so we walked for hours and hours, just basically rubbed our feet to the bone, and no food, no lights, nothing. And finally stumbled into camp the next day at 6 o'clock in the morning. Uh, really, really mad.
Where in Ecuador was that?
It was I don't want to
Okay.
make anybody feel too bad. It was in the northwest,
Copy.
Esmeraldas
Yeah,
province.
I was gonna say, depending on yep. Yeah, yeah, that's no, it's a beautiful forest.
Uh-huh.
And some of the most biodiverse areas. I was just in Ecuador last year for a couple months and kind of all over. but it was beautiful to be back in that area.
Yeah.
And I
I don't know
know
how it works for you, but for me, I'm very opportunistic in where I go. It's a
absolutely.
it's a combination of where we can get permission and where it's somewhere that people haven't been before. That's my preference to go, to make new collections.
Yeah, or underrepresented in collections.
Yeah.
Yeah, for me, it's a mix, and it would certainly fall into those categories. A lot of what I do is facilitating the access and permissions for certain locations, particularly that are understudied and also threatened in a variety of ways. But, early on in my career, it was very opportunistic in terms of um, to be completely honest and fully confess, doing whatever it took to get to the tropics in terms of I would study anything, I would work in whoever's lab or write grants on anything that I felt like would be successful just to get into the field. And that went, that ended up spanning for a while, just gaining experience, writing proposals that, you felt like you would have a chance just to get away for the summer or a semester or something like that. And that ended up spanning butterflies in the tropics or primates or who knows what, soil ecology in the Amazon or coral reefs in Africa. So definitely opportunistic for a long time as I was trying to just at that stage where you're just trying to gain experience. And then it later became more of the facilitating logistics to the extent that you're talking about of where can we get permission to go, um, and focusing on that on the more difficult to access locations because there is more of a niche in that type of work. But and you've also worked outside of the Amazon. I know you've also worked in Africa and Southeast Asia as well.
Not in African rainforest, no, but in Thailand for sure.
Yeah.
And I've been to other rainforests in Southeast Asia. For me, the mountainous areas, the Himalayas, are more interesting in Asia than the lowlands. It seems like the diversity changes every valley practically.
Yeah.
But there's lots that's not known. And a lot of the diversity that we work with, of course, is cryptic. So unless we're out there sequencing these things, what looks like one species that's widespread isn't necessarily so.
Can you elaborate on that a little bit for maybe people in the audience that don't have as much or aren't in this specific field about cryptic diversity, cryptic species?
Well, it's becoming more and more obvious to me that what we think of as widespread kind of variable species are in fact complexes of many species, and not always, but sometimes. And the way we figured this out is by looking at their genes, specifically at the cytochrome oxidase 1 barcoding region. That's a mitochondrial gene that provides a sequence of bases that is usually distinctive for a species. And that's not to say it doesn't vary, but the variation is quite low within species. And once we start looking at this, we start getting things like a species that I've been working with in North America and South and Central America through the New World is now 16 species. So once you start adding those things up, your estimates for the number of species in the world starts to get really high. And a recent paper has estimated that there's 168 million species of animals in the world, based in large part on this sort of cryptic speciation of insects that has, well, I wouldn't say that cryptic speciation has taken place. Speciation has taken place, but it's taken us some greater effort to detect it. So what we do now is we get specimens or parts of specimens, and we soak them in an extraction buffer that takes DNA out of the muscle tissues, and then we sequence them to figure out the actual pattern of base pairs in their CO1 genes.
Perfect. That was a great description. Um, and that actually leads me, I mean, from the work that I'm doing now with XPRIZE on this XPRIZE Rainforest about innovating and advancing, revolutionizing tech for the survey of biodiversity, monitoring biodiversity, to more, rapidly and autonomously assess the tropical rainforest now and inform conservation action and policy in real time. Um, how do you see? I know that the advancements in how we are able to genetically study species in the past 10 or 15 years has really skyrocketed. But how do you also see, and maybe in the work that you're doing, advancements in various aspects of technology? How do you see that changing the way that we approach the work that we do and conservation or studying biodiversity in the tropics? Are there specific ways in which you're seeing the field moving forward because of technological advancements?
Well, certainly through sequencing, it's going to be possible. I don't know if it's advisable, but that's a highly debated subject. I think it's advisable to go in and describe huge swaths of biodiversity based mostly on a barcode and a couple of photographs. Because really, these groups are so huge, we don't have time to sit down and do a careful description of every body part and illustrations and so on. We'll never get the job done.
Well, and it even goes back to, I mean, the traditional way that a lot of this work has been done, the way that we've done it for years, the way that museums and researchers have done it for years. Like you said, I think individuals like us are so used to being in the field all the time, gathering data. And it's interesting for you to say that the pandemic has allowed you to actually start to catch up on the backlog.
Mhm.
But if you think about collections like at the Natural History Museum in LA, um, all of the information that's stored there, and we've had great conversations in this series in the previous uh, episodes or whatnot. But there's so much information that we as humans don't. The way that forests are being degraded and compromised, and the way that the world is changing is so urgent that we certainly need to escalate the forms in which we can assess biodiversity, analyze large amounts of data, because I think there are times when we're processing samples from collections collected decades ago.
Which
Um,
has value, of course, when you're looking at change over time.
Absolutely, but there's also areas that I'm sure you and I have been to. I know I have for sure that by the time you're analyzing certain things, or by the time you return to certain locations, those forests may no longer even exist, or they're severely altered. And so, though certainly I think the way we've seen sequencing progress in the past 15 years or so, from you talked a bit about CO1 barcoding of one gene to multiple genes to whole genomes, and even the cost of that has come down substantially and become far more affordable. Um,
Right.
uh,
But it's a lot more work and money too to do that.
Absolutely.
Yeah.
Yeah. And that, there's certainly something that we could all use more of is funding for science and research.
Yeah. Well, studying biodiversity for conservation purposes is very different than studying it for systematics purposes. Systematists tend to go out and cherry pick new things, or what appear to them to be new things. Whereas if you're trying to figure out how important site A is versus site B, you need to do a more comprehensive sampling. And the malaise trapping with barcoding allows you to do that much more easily. It's not easy. It takes a lot of effort and just technical work. Somebody has to pick each specimen up and put it in there. Another approach is you take the whole sample and you blenderize it and you sequence it and you sort out the different barcodes that you get out of it. It's called metabarcoding. but then you lose all the structural information, what the flies or what the animals looked like and how many of them were in there. Basically, you can't tell that from a metabarcoding sample. So human labor is a large portion of this. The good thing is that untrained labor can do a lot of that work, loading up the cells, and then the rest of it's just chemistry. Except, well, just chemistry to get the barcodes. And then somebody, preferably an expert, has to look at the clusters of barcodes and make sure that they correspond to what we consider to be species.
Well, and even that in itself is a very confounded subject these days. Certainly with the advent of the type of analyses of genetic data that we're talking about with cryptic species and everything like that. What is a species has become more and more nebulous with certain groups, especially when you get into plants and things like that. It becomes a nightmare. But one thing I want to just point out to the audience as we're getting to this stage in the conversation, we're happy to take questions both on the Zoom platform, or I believe we're live on YouTube as well. And we might be able to field questions from there also. So feel free to, I guess, send them in through the Q&A function on Zoom or chat or the comments section.
So Peter, what would you say is the richest place you've ever been in terms of biodiversity?
Richest place. I mean, a lot of people talk about the foothills of the Andes in the Amazon, where we've talked about in parts of Ecuador around Yasuni and parts of Peru. You mentioned the Himalaya, and I do feel like areas where you have elevation gradients, I'm very interested in endemism. And so a lot of my work has been on islands from Borneo to Madagascar, and even in the Americas and islands throughout Central America or the Caribbean. And I have a particular fascination with that, island biogeography. But, some of my early work, while I did a lot in Ecuador and the Amazon and other parts of the Amazon, I've always had a special place in my heart for Borneo. And so that stands out. And I've worked a lot in Madagascar, where everything there is, all of the biodiversity is unique. So I have a very difficult time answering questions like that. I mean, objectively, in a sense, parts of Ecuador, Colombia, Peru are accepted as most biodiverse places on the planet for certain taxa. And as well, I kind of have personal favorites that come and go throughout, depending on where I'm working at the time. I have
favorite places are different than most diverse places.
Absolutely. And I would say right now, some of my favorite places are in the Congo Basin. And I think a big part of that comes also with the level of work that is still yet to be done in terms of study and understanding the many facets of biodiversity in that region, which happens to be the second largest rainforest in the world. What about you?
For me, you can't beat the areas around Manaus and Iquitos and Leticia. Those are the, central Amazon.
Yeah.
Those are the places we've gone and actually run out of vials. We got so many things. Or we go into the field and we're looking at different interactions between a species of ant and the fly. And we have to triage the whole day, basically. Oh, I really want to study this, but I've got to look at that because I've never seen it anywhere else.
Yeah.
So those are the places.
Yeah, Leticia. I'm itching to get there. But on that note, I know that you take photos as well, and I being a photographer myself, what do you? We chatted a little bit about this recently, but there's that fine line between getting the research done and doing your science and being able to take photos. But when you're able to take photos, what do you enjoy most about doing that on your trips?
Photographing new behaviors, basically. We found a new species of fly in Brazil that was the actual female flies were chopping the heads off the ants and carting them off into the forest. So that was a totally new behavior. It's a second type of ant decapitation in the forest, because the normal type is the female lays their egg and the larva feeds in the ant's head and the head falls off. In this one, the fly actually cuts the ant's head off and carries it away. So those sorts of things, documenting new behaviors, is really a lot of fun, especially because it's so unexpected.
Absolutely. I feel like there needs to be, we should talk to different colleagues. I could talk to natural history filmmaking colleagues about, a whole savage series of insect interactions that's entirely macro lens filmed, honing in on these little warriors in the jungle.
Yeah, life is brutal when you're an ant. Yeah.
That's what we still have a few minutes left, but that would have otherwise been a great line to end on. Life is brutal when you're an ant. We got one question from the audience here, and while I read this, I want you to also think on that note. I know we've talked a little bit about parasites. You work on parasitoids. We could certainly dive into the whole parasite question on humans. I'm sure we both have some gnarly experiences in that, but anything that comes to mind for you?
Oh, I have a story from being in the, have you been to Paquitza in Peru, outside of Manaus?
Yeah.
I was there one time walking down a trail with my colleague, and he turned to me and said, "Brian, I think I've got a botfly maggot in my scrotum."
Oh.
Which is not something you hear every day.
Hopefully not every day.
You know what botfly maggots are like. They're these maggots that get to be about this big, and they have spines all over their outside. And so the fly actually lays its eggs on other flies. Then the fly lands on you, usually a mosquito or some other kind of biting fly. The eggs hatch and the larva goes into your skin. And then the larva starts sort of spinning around, lacerating tissues, and that causes infection. But that's good, because that's what the fly larva eats. It filter feeds out the bacteria in the wound. So in order to continue this process, it's always spinning around and spiking you. So anyway, you know it when you have one of these larvae in you. You've happened before.
I've had many, but never there, fortunately.
Right.
Gosh. The first time I got many the first time I went to French Guiana. And there are certain sites where certain, tropical, like infectious diseases are more prevalent than others, or certain times when there are certain outbreaks. And likewise for certain parasites, there are certain field locations, certain forests where botflies are more abundant and more likely. And unfortunately, you kind of just know that you're going to come out with a number of them. And I was in the field in French Guiana, had a couple, but then I had another one as I was, at the time I was based in Florida. And I remember flying to Miami, renting a car. And as soon as I got on the highway, we were talking about this previously, but for those who have never had a botfly before, who have never had the pleasure of this experience, it's a very startling, it feels like you're being jabbed with a knife periodically, but it's very sudden, it's very unexpected. And I remember driving, it was actually an entomology trip, and we rented a car, we're merging onto the highway, and I felt this jab I kept touching my arm even as I was subconsciously telling this story. That one was on the inside of my arm, and I nearly swerved off the highway because it was so unexpected. And it's just quick, but over time, over that period of about a week or two, the frequency increases. You get woken up throughout the night every 30 minutes or so. And yeah, but that one in particular stood out, and there have been many throughout the career, but yeah, certain ones like that.
Well, my friend didn't have a botfly maggot, as it turns out. We had a doctor on the trip, and the doctor was able to remove it. You know how you have to put something over the breathing hole and the larva comes out far enough to pull it out. So he pulled it out, threw it in a vial of alcohol, and said, "Nocho, another one." Pulled it out, another one. Pulled out 19. And so he gave them to me, and I looked at him and said, "No, these aren't botfly larvae." It turned out to be primary screwworm larvae, which actually attack healthy tissue, unlike the botfly larvae, which just suck up the pus, basically. And my friend had a total of 22 removed from him.
That's suboptimal.
Yes, suboptimal is a good way to put it.
Man.
But there's a happy ending because he was able to reproduce. No permanent damage was done to any important organs.
Well, that's one kind of, yep. We're trying to keep this as, there's so many things I could say on that I'll avoid saying, but one thing I will say is the glamour of working in the tropics is not what people think it is. That's one good way to sum it up. And I would always say, a fascinating part of my job in terms of leading field teams and leading expeditions is monitoring individuals, and making sure that your teams, that you have that relationship with people and that respect and trust and like understanding, so that people do open up about things, not necessarily to that extent or that kind of scenario, but even, mental health or whatever. But when you're in the field in tropical rainforest, it's a very strenuous, demanding, challenging environment in many different ways. And so having that, that bond between people and being able to monitor people in a way to make sure that you're on top of all of these different things that may come up, let alone, relationships with different individuals on the team and team dynamics and challenging situations. But yeah, the bot flies, you remember those for sure, as along with all the other parasites that have also joined the expedition along the way.
Well, most people who are uninitiated in tropical field work worry about snakes, right? That's the number one thing.
That's a good point. And to be honest, I mean, I personally love snakes, and so but at the same time, I have had trips where you bring certain herpetologists on, people who are, world experts in reptiles and amphibians. And there are certain locations that are very difficult to access, that are also difficult to extract from and get medical attention. Where that's the thing that you're not going, snakes in a natural, just like living in the field thing, are not the threat or issue normally that people may think they are, but it's people that are actively handling venomous snakes that then suddenly become a source of anxiety for me, even though I love to see the diversity of snakes in certain areas.
I tell people they should worry about ants, not snakes.
It's insects, really. Yeah, it's the thing, and also things like, mosquito-borne illnesses and things like that inevitably over time working in the tropics, you unfortunately tally up a number of those types of things. But yeah, it's, for me, one aspect also of working on islands that I like is being able to camp just within the forest adjacent to the beach. So at the end of the day, you could at least go for a swim, or in certain areas, maybe like in the Amazon, or for me, there are certain spots in Borneo that I really like to come out of the field at the end of the day and go for a swim like in the river, and do something that's refreshing, because so often it's so hot and humid, and I think we get used to it, or maybe we're just individuals that have tolerated it, or have a certain tolerance to continue doing it. But at the end of the day, they're beautiful environments, and I think our passion for that biodiversity in these areas, just you endure so much because of your love and passion for understanding these environments and protecting them. And with that, I really just want to say thank you for such a wonderful, all-encompassing conversation. We could talk all night about all sorts of different things, and certainly we were getting into some really gripping stories. But I thank you for your time, and I don't know if you want to say, I would just like to say once again, thank you to the Natural History Museum in LA for the partnership with XPRIZE. Our Rainforest XPRIZE early registration is still open until October 15th, and regular registration goes until March 15th of 2021. You can find out more about that at rainforest.xprize.org or email us rainforest@xprize.org. Brian, any last comments?
Yeah, I hope that people who are interested in the conservation of wildlife and biodiversity will consider giving money to organizations that preserve tropical rainforests. You get an inordinately high bang for the buck by supporting them, just because so much of the world's biodiversity is concentrated in tropical forests.
Absolutely. And now more than ever, I mean,
Yeah.
UN reports just came out about missing a lot of all of the key targets for biodiversity for the past decade. And so any support that people can provide at this time goes a very long way. And with that, Brian, I just want to thank you once again. I look forward to our continued conversations about all sorts of aspects of rainforests and biodiversity. And I thank everybody for tuning in tonight. And we will have our final, at least for this series of Tropical Thursdays, will be October 15th. So be sure to look out for that. And until then, I hope everybody hangs in there. And yeah, we'll see you then. Thanks again, Brian.
Thanks, Peter.